Welcome to this explainer. Okay, I want you to imagine for a second that you are a detective. But your suspect? It's not a person. It's actually a microscopic killer. And your victims? They're wild animals, roaming through dense forests, trekking across jagged mountains, or hiding deep in underground burrows. So how on earth do you track a culprit that you literally can't even see, on a victim you can't even touch? Today, we're opening up the case file on one of the most devastating wildlife diseases on the entire planet, Sarcoptic mange. Specifically, we're looking at exactly how Sarcoptic mange is diagnosed in wildlife.
Because that question right there, that is the core problem wildlife scientists and conservationists are facing every single day out in the field. I mean, think about it. If your dog gets sick, you just take them to the vet, they sit on the exam table, and they get tested. Easy, right? But a wild, elusive fox, a 200-pound black bear, there's no way. Capturing these wild animals just to get a medical sample causes them immense and honestly sometimes life-threatening stress. So scientists are constantly walking this incredibly fine tightrope. They have to perfectly balance the absolute need for hard, accurate scientific evidence to confirm a disease with the critical, non-negotiable importance of maintaining animal welfare.
Okay, let's dive into this case file, which is actually based on a massive systematic review of 228 scientific studies. Our agenda for today is simple. 1. The threat of mange. 2. Non-invasive detective work. 3. Gathering hard lab evidence. And 4. The ultimate diagnostic strategy.
Starting right off with Section 1, the threat of mange.
Now, to really understand why diagnosing this disease is so absolutely crucial, you've got to realize the sheer scale of the devastation here. Just look at this number. Sarcoptic mange has the widest host range of any terrestrial parasite. We're talking about at least 148 mammalian species across 12 different orders worldwide. That is huge. It hits everything from mountain gorillas and cheetahs to black bears and wild boars. And when an outbreak hits what we call a naive population, basically a group of animals that hasn't ever been exposed to it before, the results are catastrophic. For vulnerable species like bare-nosed wombats in Australia or those endangered kit foxes over in California, a mange outbreak can literally push a fragmented population right down into an extinction vortex.
The culprit behind all this is Sarcoptes scabiei. It's a highly contagious mite. And these mites, they don't just hang out on the surface of the skin. No, they vigorously burrow deep down into it, laying eggs and leaving behind allergens, stuff like saliva and feces. As you can imagine, this triggers an intense, agonizing irritation for the poor animal. They lose their hair, their skin thickens up and cracks wide open, and they end up burning just massive amounts of energy trying to cope with the itching. Ultimately, the saddest part is that severely infected animals don't even usually die from the mites themselves. They actually die from starvation and secondary bacterial infections that sneak in through their broken skin.
Which brings us to Section 2, non-invasive detective work.
So because catching these fragile animals is so dangerous for their health, our scientists' very first line of defense is gathering clues from a safe distance. They've got 5 main non-invasive tools in their surveillance arsenal. Visual observation with binoculars is kind of the go-to. Then there's spotlighting, which helps researchers find nocturnal animals in the dark. Camera traps are awesome because they're motion-activated and can watch the forest 24/7 without a human even being there. We also use infrared thermography to spot heat signatures from inflamed skin. But honestly, the most fascinating tool? Detector dogs. Listen to this. In one amazing study in the Italian Alps, trained Bavarian mountain hounds used their incredible sense of smell to track down 63 live mange-infected animals over 15 years with 0 false positives. 0! But, crazy enough, despite being that accurate, detector dogs are still rarely used because they require so much specialized training and upkeep.
But, there's always a catch, right? While watching from afar is fantastic for keeping the animal's stress levels low, it has a pretty major pitfall. If an animal has super thick fur, or if the infection is just starting out in its early stages, the physical lesions are completely hidden from view. That means researchers looking through those binoculars or scrolling through camera trap photos might actually misdiagnose a sick animal as totally healthy. So, while these non-invasive tools give us a really great big-picture view of population health on their own, they just aren't enough for definitive, foolproof disease confirmation.
Now, what's really interesting about this slide is how it shows the reality of what's happening out in the field. Out of the 228 studies analyzed, visual observation leads the pack. It was used in 141 of them. But look right behind it. Skin scraping is right there, used in 133 studies. So what does this actually tell us? It shows that scientists heavily rely on transitioning from the most common non-invasive method, just looking at the animal, straight to the most common invasive method, getting a physical scraping of their skin. They need that hard proof.
Let's move to Section 3, gathering hard lab evidence. Welcome to the laboratory, everybody.
Let's quickly contrast these two buckets of diagnoses. Non-invasive methods mean low stress for the animal, and you can study a massive population at once, but your accuracy takes a hit. Invasive methods totally flip that script. You get incredibly high diagnostic accuracy on one specific animal, but it comes at the heavy cost of high stress from capturing and restraining them. It's this incredibly cruel irony, right? To definitively save an animal or protect its population, you kind of have to subject them to immense stress first. And that is exactly why invasive methods have to be strictly, carefully targeted. We can't just apply them blindly to every single animal in the forest.
And that brings us to our heavy-duty laboratory toolkit. There are 4 main invasive techniques here, and they all require capturing the animal and using some pretty specialized, often expensive lab infrastructure. First up is skin scraping, where researchers physically collect crusts to spot the mites under a microscope. Then there's PCR, which actually amplifies the mite's DNA. We also have ELISA, which is a blood test looking for the host's immune response, specifically their antibodies. And finally, histological analysis, which basically means taking biopsies to look at microscopic changes in the tissue.
Now, even though skin scraping is the most popular invasive method, it's definitely not perfect. In fact, its sensitivity can drop to just 50% if the mite count on the animal happens to be low or if the infection is super new. Plus, it relies heavily on the skill of the person doing the scraping. If they don't scrape deep enough, or if they just happen to pick the wrong tiny patch of skin, they'll miss the mite entirely. Boom, false negative. That's exactly why scientists often have to dig even deeper, right down to the molecular level.
When researchers need absolute 100% certainty, they turn to PCR. Here's how it works. Step 1, they physically extract the mite from the skin sample using isolation or flotation techniques. Step 2, they amplify its DNA. Finding the mite's unique genetic footprint is an incredibly powerful tool. Not only does it absolutely confirm the disease, but by sequencing that DNA, researchers can act like true detectives and trace exact transmission pathways. For instance, in the Aosta Valley in Italy, researchers used DNA microsatellites to figure out if mange was being passed between predators and prey, specifically between red foxes and wild boars that were sharing the exact same habitat. How cool is that?
Then we have our serological testing, like the ELISA blood test. This one looks for antibodies against the mite in the animal's blood. It's a fantastic way to detect asymptomatic carriers or just to understand the historical spread of the disease through an area. But it has a pretty major nuance you need to know about. An animal might have caught mange, fought it off completely, and been totally cured, but they'll still test positive on an ELISA test. Why? Because those antibodies stick around in their bloodstream for a long time. So while it's highly sensitive, it can definitely throw some false positives if you're only trying to find active current infections. Bringing us to Section 4, the ultimate diagnostic strategy.
All right, after synthesizing data from over 200 studies covering tens of thousands of animals all over the globe, what's the ultimate master plan here? So the crucial point is this tiered, 3-step strategy, highly recommended by researchers, to perfectly balance animal welfare with scientific certainty. Step 1. Start at the population level. You conduct broad, non-invasive surveillance, think camera traps and visual spotlighting, to monitor the whole ecosystem's health without stressing a single animal. Step 2. Suspect identification. You use that broad data to pinpoint the specific individuals that actually look sick or are acting strangely. And finally, Step 3. Definitive confirmation. You only capture those highly specific, targeted individuals to perform your invasive lab tests, like skin scraping or PCR. It's a brilliant way to balance minimal interference with maximum diagnostic accuracy.
Which leaves us looking forward, right? We know what works today, but what about tomorrow? As we keep pushing the boundaries of technology, how will advanced, long-range tools revolutionize wildlife disease detection? Could we be looking at a future where AI analyzes drone footage in real time, or where we use next-generation environmental DNA swabbing? Could we actually get perfect, stress-free diagnoses without ever laying a finger on the animal? It is an absolutely fascinating frontier for wildlife conservation. Thank you so much for joining me on this explainer, and I will catch you next time as we continue to decode the incredible science of our natural world.